Dando continuidade aqui no evento. Boa tarde, meu nome é Cláudio, eu sou estudante de pós-graduação em genética e melhoramento de plantas aqui da UFLA e representante do GEM. Eu irei dar início à segunda palestra sobre interação genotipo por ambiente. Dando continuidade, agora a gente vai iniciar a palestra do Dr. Fred van Eeuwijk, que possui graduação em Biologia pela Utrecht University, na Holanda. Foi estatístico na Plant Research International e é professor associado em Estatística e em Melhoramento de Plantas na Wageningen University. E desde 2006 é professor em Estatística aplicada pela mesma universidade. Suas pesquisas incluem análise estatística da interação genótica por ambientes e desenvolvimento de metodologias para o mapeamento de QPL. Now, I give the floor to Dr. Fred, who will give a lecture entitled Genotype by Environment Interactions, Old and New Perspectives. Welcome, Dr. Fred. And please, could you share your slides?
Can you see it?
Yeah.
Okay, I'll put it presentation mode. Okay, so when the image and the sound is okay, well, this is a very general title that allows me to say quite a number of things. So basically, I think over the last year we've been thinking a lot on what to do with environmental information, also secondary phenotyping information coming from phenotyping platforms. And in this talk, I want to say a little bit about, let's say, some ideas we have about environmental characterization and how to define the environment in such a way that is useful in the prediction of genotype and environment interaction. Now, if we take as a starting situation the standard two-way analysis of variance model, we can elaborate that in an analysis of variance context in which all the parameters are fixed, which mainly means that we assume that we don't know anything about them beforehand, and that they can be anything between plus and minus infinity, or when we accept a mixed model formulation, actually what we do is we make some assumptions that the genetic effects behave according to a normal distribution. So if you look at a number of old papers, then you might observe that many of them start with, well, let's say, telling you that genotype and environment interaction is a problem and you can ignore it, avoid or exploit it. And funnily enough, if you just type in ignore, avoid and exploit in Google, then you find about 300 of those images about risk management. So apparently, in the past, genotype and environment interaction was seen as a risk. And my point is that actually I think genotype and environment interaction is always present, because whenever you're going to test a set of genotypes in particular conditions, you should be aware that when you slightly change the conditions, that the phenotypes will look different. So at all times, you would like to be able to model G by E, and I say only when you have strong indications that the G by E is not very large, you might decide that you want a simpler model. Now, if you look at the literature, there are many methods, many models, and let's say you might get confused by all these options that you have. But my idea is that you'd better search for a kind of modeling philosophy that allows you to use all of those methods in a decent and straightforward way. And I think then in principle, G by E doesn't need to be such a big problem, and you can easily mitigate the risks that seem to be attached to G by E. Now, the title of my talk was actually inspired by the fact that when you think about G by E, actually you can always look at it from two sides. You can look at it from the point of view of the genotype, but you can also look at it from the point of view of the environment. And when you think about the genotype, well, you might be thinking about how to characterize those genotypes. You can think about adaptation in general. How are they doing in a certain condition? You can think about adaptability. How do they change when the environment changes? You can think about stability. Does anything happen when we clearly change the situation? And resilient, well, is a more fashionable term nowadays for what used to be stability. Now, if you think about the environment, I think many of you, and also in the former talks, in the lightning talk and also in the talk by Lucia, we already saw that we always think about a kind of target population of environments. I will say more about that later. And we try to make life simpler by cutting up the target population of environment in a number of adaptation zones or ecological zones or mega environments, so that our modeling becomes simpler and also our inference, let's say that is our prediction. Now, I think it's also useful to think about G by E from the point of view of prediction and of understanding. Now, I have to say that these two are not so easily separable. They tend to form the extremes of a kind of spectrum. But when we think about prediction, in a sense, we don't care too much about the kind of model or the structure of the model that we're using. And genomic prediction, I think, to a large extent, is a kind of, well, black box approach to well, genetics and also G by E. On the other hand, when you want to emphasize understanding, the structure of your model matters, and the parameters in your model should have an interpretation, and let's say they should give you a grip on the situation. And I think in that sense, it's important that sometimes you read about repeatable and non-repeatable G by E. I would say repeatable G by E is a kind of G by E that you can reproduce, you can intervene on it, and you can change the situation, you can change the management in such a way that, let's say, you can, well, more or less decide that your phenotype will have a certain value or form. And if we're thinking about understanding our phenotypic traits, well, I think we need more complex models. We in a sense, need multi-trait models, multi-environment models. We need to see the dependence on time. We also need to see how the traits influence each other, and also we need to figure out whether genetic influences are direct or indirect. So I think to a large extent, if we want to understand how genetic correlations come about, well, we need to do more biological modeling than we typically do in plant breeding. Now, if you're a, let's say, student in plant breeding and you ask yourself, well, what should I know about statistics? Well, if you read these slides, you might get a little bit disappointed because it looks like you have to be a statistician to be a plant breeder. Well, I don't think that's true. I think that if you look at the first 3 types of models, you can, well, answer quite a number of questions, and everything that comes after the first 3, well, is nice. But in a sense, you'd better try to figure out whether there are experts around you that can help you with this. If you can translate your plant breeding question in a statistical question, then all of these more advanced methods might be useful. But most of the questions, I think, in principle, can be answered with the first 3 types of models. So let's have a look at those models. I mean, I write down here kind of mixed model, genotype I, environment J. There's an expectation, and the expectation is typically, well, it's fixed in this case, which means that we would be able to reproduce it. It's based on known covariates for the environments and the genotypes. And for the genotypes, it's quite often markers, candidate genes. For the environment, well, it's the major drivers of G by E. This is a random G by E term, can also contain still main effects, genotypic effects. We, let's say, typically don't distinguish too clearly between the main effect for the genotype and the G by E. Now, this is random, but still, if we know about the correlations between genotypes and if we know about the correlations between environments, we might still use this random genetic effect to do some predictions for new genotypes in new conditions. This kind of residual, epsilon, well, we can also do quite nice things with that in terms of spatial modeling, etc., but we won't talk about that today. Now, I think an important issue here is that most of what we try to do in the modeling genotype by environment interaction is to develop what we call separable models. And separable models is nothing else than that we try to predict the performance of genotype I in environment J from a model that contains parameters that only depend on the genotypes over here, these theta i's, or only on the environments, these theta j's, or let's say we can use genotypic covariates indicated by X, or environmental covariates defined by Z. Now, if you want to predict, you want to use as many covariates as possible and estimate as few parameters as possible. If you want to understand, you're willing to pay a certain price in the sense that you're willing to estimate more parameters. I mean, that's, I think, something that you need to take into account. But basically, our modeling challenge is what you see on this line over here. For the part that you can manipulate rather simply, you want genotypic and environmental covariates, and you want to estimate the parameters related to the genotype and the environment. If you don't want to distinguish between the parameters and the covariates, you can write, let's say, the performance of genotype I and environment J, you can write as a product. A is referring to genotypic sensitivities, B is referring to environmental characterizations. Here are a number of examples in which, well, here you have, for example, a marker covariate. X stands for markers, and beta, well, is then something that you estimate in the environment and can be a QTL effect. So the QTL effect differs going from one environment to the next. And you can have a number of those. That's why we have this summation sign. The other way around, you can have environmental characterizations, for instance, temperature or a drought index, and then you estimate sensitivities of the genotype with respect to the drought index. You can have products of genotypic covariates and environmental covariates, and then you only estimate a kind of proportionality constant. This kind of formulation is underlying, for instance, this rather advanced model that Diego Cartina is using for genomic prediction. But then you have many of them and you penalize those parameters because you cannot estimate all of them. If we go to the random part, actually that is the part that we cannot produce so easily because we don't know very well what it is that is driving the genotype, and let's say to what the genotype is referring the environment. Well, you get a formulation that I won't explain here, that in essence has the same property of being separable. It depends purely on the genotypes or purely on the environment, and the product of those two allows you to predict. And what kind of information can you use? Well, for the covariates on the genotypic side, we have markers, high throughput genotyping, we have pedigree information. We can have phenotyping platforms, we have high throughput phenotyping information coming from platforms. And for the environment, well, you saw it already in the talk by Nathalie, and all kinds of ways to characterize the environment. In a little bit more detail, here are a number of models that you might know. This first set of models is a so-called bilinear models, and the bilinear models have as a property that you need to estimate genotypic sensitivities and environmental characterizations simultaneously from the phenotypic data that you're analyzing. And so that can be yield. Well, then delta is a theoretical characterization of the environment based on yield, and gamma is a sensitivity, a genotypic sensitivity related to that theoretical environmental characterization. You can also have what we call factorial regression models, and these were shown in also in the slide or in the presentation just before me. Once again, this could be a marker, and here this could be a property that you measure on a phenotyping platform. For instance, capacity to photosynthesize, and then beta is the amount of light that comes in. So the capacity to photosynthesize times the amount of light and the resource provides you with biomass or yield. Well, you can do the same for the environment, and this set once again is referring to the environment, could be a stress index, and in this case it has index J, which means that the stress index is defined for all the genotypes simultaneously for a particular environment. You could also have the stress index genotype and environment specific, so then each genotype experiences a slightly different type of stress in a particular field condition. Okay, and then, well, we can have these products of genotypic and environmental covariates, and yeah, typically you can think of that the genotypic sensitivity is partitioned in a part that you can directly relate to a genotypic covariate and some residual sensitivity. Okay, so now if we think about genotypes, well, actually we could characterize our genotype by a set of genotypic parameters, the A's. If I think about the environment, I could think about environmental characterizations, and this can be either estimated or measured as covariates. And if my system is well known and I know which are the main environmental drivers, then the values on those environmental covariates could be enough to characterize my environments. However, if I don't know so well what's going on, I might prefer to look at predictions from a GxE model, and try to characterize the environment by, well, the predictions for the performance. If I'm more interested in understanding, I could look at the estimates for the genotypic sensitivities together with the estimates for the environmental characterizations and try to figure out whether I can define a model that embeds those effects and also in a way that we call conditional independent, which means that nothing else is going on except the relationships that I see. Okay, now I think by now it has become clear that there is certain complementarity between the genotypes and the environments, and if we write the models as separable models, genotypic sensitivities or scores times environmental characterizations or scores, then we understand that I can only describe my genotypes well if I know the major environmental drivers. That's a bit curious, because if I know everything, yeah, okay, then it's obvious that I don't have any trouble estimating the genotypic sensitivities that matter. The problem, of course, is a little bit that typically we're not completely sure what our target population of environments is. I come back to that later. To understand the environment well, actually we need to know to which type of stresses our genotypes will respond. And if my genotypes, well, differ in drought tolerance, well, actually, then I need to expose them to drought tolerance. Otherwise, I will never know which ones do react and are sensitive and which ones are relatively tolerant. And let's say if my trials are, well, majorly differentiated by a particular type of stress, then to find out what that stress is, yeah, I will need genotypes that are differentially sensitive to that stress. So the choice of genotypes and environments, actually the training set of genotypes and environments for my particular GxE model determines to a large extent whether I will be successful extrapolating my results to new genotypes and new environments. Now, maybe it's good to say that I think when you look at how we try to solve those GxE problems, plant breeders and geneticists are very strong in looking at the genetic differences, but probably physiologists are better in understanding the environmental differences. And I think by now, with all the new data coming available for genotypes, phenotypes, and environments, I think increasingly we need environmental information to build our GxE models. And so a kind of balanced approach is what is required. For the target population of environments, well, by itself, I could easily spend a whole hour talking about different ideas for this target population of environments, but in a nutshell, it's more or less the total set of growing conditions, future growth conditions that our genotypes might meet in the future. And Nathalie already gave, I think, an elaborate list of, well, various covariates that determine the physical factors that determine the growing environment, and of course beyond that there are also all kinds of biological factors that actually you would need to take into account as well. I think in the GxE case we tend to model very strongly the abiotic stresses. We don't do so much for the biotic stresses. Now, if I would think about climate change, how am I going to prepare my future cultivars? And then the first thing I need to think about is, well, for what kind of environments and am I developing my genotypes? So what I need to do is I need to insert genetic mechanisms that provide tolerance to, well, for example, drought stress, if this is the major stress defining my future growing environments under climate change, probably also heat stress. And to identify such genetic factors, well, I need to test my genotypes. Now, how am I going to do that? Well, the strategy in which I create multi-environment trials, combinations of sowing time, year, location, and I hope that the right type of conditions does occur for selecting the right type of genotype. I can also try to understand what's going on in a more physiological way and impose particular stress on the genotypes in the hope that the ones that I select will also do well later in the field. So the question, when I depart from this simple model of sensitivity times environmental characterization is, well, can I estimate genotypic and environmental characterizations that help me to predict the G by E for my particular target trait? Now, a general strategy to simplify life and breeding is to try to group trials, in a sense to cut up the target population of environments. And this can be done by scenarios. This is quite popular in relation to climate change. And the idea is that a particular combination of soil and meteorological conditions creates a unique environment, and those unique environments are defined in classes that we call scenarios. If I think more about the location where I am, the soil conditions, I could try to cut up my target population of environments in regions, and these are, well, next to each other. If I don't care so much about whether my, well, trial locations, my future target population environment, whether it consists of locations next to each other, I could also look at mega environments or ecological zones, which are also, let's say, defined in space, but don't need to be continuous. So, okay. I'm going to make groups of trials. I can look at multi-environment trials, as I mentioned already, and I can try to group the trials in an observed sample of trials in my multi-environment trial. If I do this well, I can explain the genotype by environment interaction or the genotype by trial interaction in terms of genotype by region or genotype by scenario. Another option is that I use my past weather records, maybe going back 100 years, I use soil characterizations and I run for a particular set of genotypes a crop growth model, and I create a simulated sample of my target population of environments. The advantage here is that I can have a more elaborate sample of the possible conditions. The price to pay is usually that I can only do this for a limited set of genotypes. Well, in terms of models, if I, let's say, start from my two-way model and I introduce locations and years, you get this three-way model with genotype by location, genotype by year, and the genotype by location by year interaction. And if I introduce regions or scenarios, I mainly, let's say, partition the original G by E in a part due to genotype by region or genotype by scenario, and a residual part of, let's say, residual trial variation, residual genotype by trial variation within regions and scenarios. Now let's look at all of this in, let's say, some examples in which I've been involved over the last few years. I think one of the most clear examples is in the work that I did together with Emilie Millet and François Dadieu. This is on maize. And here we combine a number of issues. We cut up yield in yield components. We're using marker profiles to predict yields. We look at environmental characterizations, scenarios to understand G by E. We also cut up development. We look at the growth stages to define environment-specific and genotype-specific characterizations. And we use phenotyping platforms in a smart way to understand in which developmental stage our genotypes are in the field without having to observe all of that. Two papers in plant physiology and nature genetics describe all of this work. And this is part of an EU project. And well, it's about 250 maize genotypes, more or less typical for the southwest part of Europe. At that moment, 333 cases, by now 1 million. We redid the analyses, but the differences are not very big. There were about 30 trials, 30 environments, and we had 15 environmental characterizations that actually are created using a combination of calculations and simulations. And what I think is important is we found a number of QTLs that were typical for a scenario. And the idea was, well, if a QTL is specific for a scenario, it does well, it provides yields in a particular scenario, then if a scenario becomes more frequent in future under climate change, then we know already which alleles to use. That's a very simple way of preparing for climate change. We can also have our QTLs depending on a continuous covariate. We used a rather common GWAS and genomic prediction framework for our modeling. which, by the way, was, let's say, made in-house, especially by a student of mine, Willem Kruijer, and that developed, well, possibilities to map up to till 50 to 100 traits and environments. And I have to say that the environmental characterization is very critical, because without prior physiological knowledge, it's very hard to figure out which part of the environment really matter for the prediction of yields. And the other thing is, we also think that it's necessary to know what is happening in the field with respect to developments. So the phenotyping platform in Montpellier was used to figure out where plants were with respect to the development in the field. Okay, so the model, well, I won't spend too much time on it, but basically there is an environmental intercept. This is a marker related to QTL. This is the QTL effect that is environment dependent. So each environment has a different QTL expression. This is a genetic residual that can once again be modeled if necessary by, well, marker information, pedigree information, and additional environmental information. This model produced this kind of output for yields. Here are the 29 trials. There are 6 scenarios. They are based on temperature and water availability. And where you see the horizontal lines, well, that is where the scenarios were defined. Well, here you see the scenarios, 6 of them. Here are the 10 chromosomes of maize. Wherever you see a green dot, that means that we had a QTL with the positive yield contributing allele coming from P73. And wherever you see blue, it meant that the alternative allele was yield contributing. What you can see immediately here is that our environmental scenarios actually are not terribly informative with respect to our QTL expression. It's not that some scenarios contain purely green dots and others purely blue dots, and that all of the environments or trials in a particular scenario all have the same intensity of that particular QTL. I mean, there is some indication that the scenarios are related to the QTLs, but it was not too strong. Here we look at one particular QTL. This is a QTL that did show a dependence on water availability. It had positive contributions to yields when there was water, but when it was dry, it didn't do too much. You can also see that here. Here are the 29 environments, and this is the size of the QTL effect. And when you see in the water-stressed environments, which were less, by the way, less frequent, here the QTL doesn't do too much. In the water availability, 5 minutes left only. I have to speed up. You sure? I started 2 minutes late. Okay, so here you have the, let's say, dependence on a continuous variate. This study was further elaborated. We cut it up, grain yield and grain number and grain weight. And then for grain weight, we figured out that we could explain the T by E in terms of radiation, temperature at night, and water potential. This is the model. This is a factorial regression model. And in the next step, for grain weight, we didn't have G by E. So the model for for grain yield is grain number times grain weight. And here is the factorial regression model with the main effect and the sensitivities to the environmental covariates. And those were regressed on marker profiles by genomic prediction. And so we could predict for any kind of environment using the environmental characterizations plus the marker profiles, and using a dissection, etc. Well, skip this one. Okay, so if we look further to environmental characterization, here are bilinear models. These bilinear models like Finley-Wilkinson, AMI, and GGE. Well, you can look at the parameters for the environments, but people can also look at the fitted values. This is what happens in the GGE biplot. By looking at the best genotype according to a GGE model, all trials sharing the best winning genotype belong to the same mega environment or the same environmental zone. That is one way of doing it. This is more advanced. This is using corporate models and geoinformation systems. And once again, this is a long time ago, but if you look at how the G by E is split up, you see that the environmental characterizations pick up a certain amount of the original, in this case, hybrid by trial information. And well, this is typically what we try to do. This is from Australia. Once again, there are profiles that describe, let's say, how water stress behaves over the growing season, and all trials that belong to a particular profile are assigned to the same category. And then for each location in Australia, you could indicate when those environmental types occurred, or the frequency of the environmental types, which can help you in predicting the G/E. Now, one thing that I think is very essential in recent times is because of secondary phenotyping, we have access to what we call dynamic information. And here are a number of papers and a thesis on that. And we did simulations in which we especially investigated, let's say, the dynamics of all kinds of yield components. And these are all yield components. This is yield itself. And what you see is the correlation of yield components with yields over the growing season in 3 different environmental types. No water stress, well, a little bit of water stress, and a strong water stress that is relieved at the end. Okay, so if you start to think about it, if you have access to this kind of dynamical information, it would allow you to identify environmental types by looking at the pattern of correlations. The same principle is elaborated here, environmental type 1, 2, and 3. This is yield, and these are the correlations of yield with dynamical parameters related to yield components. And we can uniquely identify the environmental types by this correlation pattern. Now, you need, I'm nearly there, you need some techniques for that. We do this at this moment by fitting those kinds of graphical models, and this is genetic information. The genetic information goes to these gray primary physiological parameters. These physiological parameters feed into intermediate phenotypes like biomass, grain number, grain weight, and finally those things need to go to yield. Now, there are direct influences, genetic influence on yields. There are indirect genetic influences coming via other traits. Each environmental type has a unique pattern of correlations, direct and indirect influences on yields that can help to tell us where we are with respect to our type of environment. And this kind of environmental characterization will help us to better predict yields. Now we'll skip all of this. So this is my almost penultimate slide. If, let's say, you're able to find environmental covariates or environmental groups that help you to predict G/E, how do I know that I'm doing well? Well, there is some mixed model technology that looks at the selection response in a divided versus an undivided target population. There's quite a number of nice papers. You can look at whether the genetic correlation between the trials in a particular environmental group are higher than in different groups. This is, for instance, in this old paper from Mark Cooper and Carlos Löffler from 2005. You can look at the commonality of the best genotypes according to the predictions of the model. And this is what's happening in the GGE biplot, but this can also be used in other contexts. Interesting, you can take a more elaborate approach in which you try to minimize the risk that selected genotypes for particular conditions fail to yield beyond a certain threshold. This integrates uncertainty and expectation. And then I think my final comment is, well, for modeling G/E, it helps a lot if you can use physiological knowledge to limit the possibilities for your environmental covariates and your environmental groupings. And I think finding a decent environmental characterization will, well, first of all, help to predict better G/E and also maybe to understand. And so this is the final slide. I think by now we are in a situation that we don't need to be afraid of G/E. I think we have enough modeling possibilities, enough knowledge, and we need to integrate breeding and physiology, some statistics, machine learning to make the best out of it. Done.
Thank you, Fred, for your talk. And now I will ask you some questions from the public.